Thank you. My name is Philippe Chambon. I'm the CEO of EG427, and it's a pleasure to be here. Thank you for taking the time. And this is not an AAV presentation, so I hope you'll find it different. Next slide. I suppose I can do that myself. Yes, okay. So EG427 was started on the base of a European collaboration between a series of academic groups that started in 2013 to find a way to generate better treatment for a devastating condition called neurogenic overactive bladder, and neurogenic detrusor overactivity. For that purpose, those academic centers decided to focus on non-replicative herpes as their tool to deliver gene therapy into the right type of tissue, i.e. the sensory neuron of the bladder there. And that's what led to the creation of EG427 in 2019. Our initial focus is on peripheral nervous system pathologies, and our first product is indeed for the treatment of neurogenic bladder, where we're at the preclinical development stage. The company is based in Paris. We have a small research team there. We are about 10 people. We've been funded with about $15 million at this stage, mostly through equity and a bit through grant also. So why is herpes vector such interesting tool for gene therapy? There are some fundamental differences here versus other platforms. The first one, to go back to the herpes virus itself, it's a very large envelope virus. It's a virus that infects 60 to 80% of human population, generally at a young age, and it stays latent in the infected cell for the life of the person. It sometimes wakes itself up and creates a small pimple on the skin, things of that nature. So it's a virus on whom we have tremendous safety history. It's a virus because of its size in which you can put very large transgene and express those. It's a virus that enables in vivo dosing. There's not a strong immune response to an infection by herpes virus, which is also why we tend to carry it through our life, and that there's no untoward effect of that. Which means that we don't need to test our patient for antibody as an exclusion criteria. It's also allows us to do repeat injection of our vector in the same patient. We can get long-term tissue expression because that's one of the inherent aspects of herpes virus. They get into their target cell, most of the time sensory neuron, where they go and put themselves in episome form next to in the nucleus of those cells. And because of the expression of a specific region of the virus, the latency region, they stay silent through our lifespan. And of course, it's a virus where there's no integration with the host cell DNA. So it has a series of tremendous advantages. The limitation one has to work around is the tissue specificity of it for the long-term expression aspect of this platform. So how do we build those vectors? We build them first starting with a herpes virus by deleting essential replication genes. In our case, what we do eliminate is ICP4 and ICP27, hence the name of the company in particular, so that it translates into a non-replicative vector backbone. And then we modify the latency region of that backbone so that we can insert there very large transgene under various types of promoter structure. We can also insert, given the size of the region, we can insert several genes with different type of promoter. And in the case of our first product, the transgene that we've inserted there is the light chain of botulinum toxin F, so that we can replicate to some extent some of the benefits, some of the advantages of Botox, but on a very local basis. Given what I've said about the herpes virus, our initial focus is on disease of the peripheral nervous system, where we can readily access those tissues and take advantage of the local effect of our vector. We can then insert large transgene to deal with diseases that are specific of different subsegments of the peripheral nervous system. There's a broad range of diseases that are accessible as potential target for our vector, and you can see the list here. We're focusing initially on neurogenic bladder, given where the company started from an academic perspective. And it's a field, peripheral nervous system diseases, where therapeutic options are still very limited. The better known of it is Botox, which is used in particular in the treatment of neurogenic overactivity. And another example is baclofen, which is used for the treatment of spasticity under certain circumstances. So with our lead program, neurogenic bladder, where are we? Neurogenic bladder arises when you have loss of central nervous system control over the micturition reflex of the bladder. When you have this, and this loss of central nervous system control can occur when you have a spinal cord injury. It also occurs in advanced stages of Parkinson's disease or multiple sclerosis, for example. When you have that, it's the sensory neuron of the bladder that take over. And the result of the activation of the sensory neurons is abnormal contraction of the bladder that can occur with or without mixture. When you have that type of pattern, you get repeated urinary infection, you get urinary leakage, and you get also increase of pressure in the bladder that over time can lead to kidney failure. So it's an extremely serious condition with a high cost to the healthcare system. From a prevalence perspective, it's a prevalence estimated to be anywhere between 400,000 and 1 million people in the developed world just for spinal cord injury. And from a market potential, it is estimated to represent anywhere between 600 to $1.5 billion, depending on penetration assumption as well as pricing in that population. We are at the preclinical stage of development of our EG-110A program, as I mentioned. And what I wanted to do is just share with you some of the key results that we've obtained in that slide. We validated now our approach in 2 key models of neurogenic diffuser overactivity. What I'm presenting here is the work we've done in a irritation model in rat, which is basically using capsaicin and infusing the bladder of the rat in those with capsaicin. The study was designed with 2 arms, a control arm and a treated arm. For the treated arm, we inject EG-110A in the bladder walls. And then after a few weeks, we infuse the bladder of those rats, the treated and the control, with capsaicin, and we measure the increase in contraction of the bladder and voiding, or if you want to look at it in the opposite way, the decrease of the interval between contraction. And as you can see very well here, at 15 and at 30 minutes post-capsaicin treatment, we see a very nice increase in the interval between contraction in our treated group, and we have nice statistical significance here, compared to the control arm, and we decrease the frequency of micturition by over 50% in the treated group. And from a safety perspective, one of the remarkable aspects of herpes vectors is their safety, in fact. As you can see here, we had, versus the control arm, absolutely no change in body weight, and we also had no change in bladder function between the two groups. This is, in fact, very much aligned with what has been seen, be it with oncolytic herpes virus that are currently on the market, or with the with the non-replicative herpes virus that are in clinical development, such as the program that Crystal Bio have. So we're working with a tool which safety is being by now extensively documented. The next step for us with this program is we're preparing for an IND filing by late 2013 to have our first in-human study at the very end of that year. We are working on our pre-IND meeting. We're making progress on our CMC, in particular on the manufacturing process, and we're going to initiate our toxicology studies before the end of this year. We're also in the process, of course, of selecting additional indication for EG110A. In parallel to that, we are broadening the usage of our platform, and this is what our research group based in Paris is focused on. And to do that, there are two key aspects that we need to make progress in parallel. The first one, we're working on the construction of second generation vector backbone. So we are continuing to modify our herpes virus, non-replicative herpes virus backbone to add to it different insertion sites, different location for those insertion sites, which would then enable us to do different transgene, different promoter expressed at different point in time and so on, which can generate some very nice change of function, change of expression function. But to enable all of this, we need to also generate the cell lines that are able to produce those vectors. So in parallel, we have an optimization of cell line effort so that we can, at the end of the day, produce those modified vectors in the most efficient way. With those type of activities, we are continuing to build what we believe to be a very strong intellectual property platform. We've already have a series of license in place based on the academic work that was done and also access to current cell line that we're working on. We filed a series of patents that covered different aspects of our virus construct. And from a process perspective, like any firm in that sector, our manufacturing process is one of the core assets that we're developing at this stage. For us to do that, of course, it's the team at EG427 that brings us forward. And I just want to mention two individuals in that team. Our VP of Research, Greg Sarek, who comes to us with tremendous vectorology experience from an AAV perspective, and also a very strong knowledge of the herpes virus family, given his work at the during his academic years at King's College. And the second person is our CSO, Alberto Epstein, who is one of the founders of the company and is one of the world experts in herpes virus biology. So with that team, we're prosecuting very effectively our preclinical program as well as the build-up of our research effort around the next generation vector. We also had the great honor of being supplemented by a terrific scientific advisory board where we have some of the key names in herpes virus biology as well as vectorology. For example, Sam Rapkin was critical to the development of the first oncolytic herpes vector virus. And David Fink was the person that brought to the clinic the first non-replicative herpes virus vector that was used for the treatment of pain. So in summary, we do believe that herpes vectors represent a very important alternative in the armamentarium of tools for gene therapy because of its ability to carry very long transgene in it and express them for the long run, and its ability also to be used on a repeated basis in the same patients. So we believe it's an extremely exciting tool. We have our first project in preclinical development in a large indication, in a large and important indication, neurogenic detrusor overactivity. We are going to be at IND stage at the end of 23 and entering the clinic shortly thereafter. And from an operating model, we're a small internal team to do our research effort, supplemented by a core of contractual organization for the manufacturing, toxicology, and so on. And we're at the stage where we're starting to discuss partnering opportunity. I want to thank you for your time.